Hello, my name is Immo Scheffler. As Professor of Biology at UCSD and as the holder of the Paul D. Saltman Chair in Science Education, I would like to welcome you to Science Matters. It is my distinct pleasure to dedicate this series to the memory of Professor Paul Saltman. Paul's passion for science was exceeded only by his passion to communicate about science. We hope to follow in his footsteps in bringing you these presentations so that scientists at UCSD can share with you the excitement of discovery and emphasize the relevance of modern biology to society and especially to you, the citizens of our community. I hope you will enjoy this presentation of Science Matters.
One of the exciting things about UCSD is that you will hear the news before the rest of the world does. One of the challenges for wildlife biologists today is trying to save disappearing species. The first thing you have to learn is how many are left. How do you count things? It sounds easy, but what if the species you're interested in is elusive and hides from you? Or what if it's dangerous and you don't want it to know you're out there trying to count it? Or what if it's discreet and doesn't want to know that you want to know whether it's a male or a female? Today's speaker, Lori Eggert, is a graduate student in the Ecology, Behavior, and Evolution section of the Division of Biology, and she's going to be telling you today about her solution to this dilemma that faces all of us. How do you study things you cannot see? How do you census elusive animals? Please welcome Lori. She's going to talk about the forest elephants of Kakum National Park in Ghana, West Africa.
As a conservation biologist, it's my goal to use scientific methods to look at the information needed to manage populations in their natural habitat. Some of the methods of basic biological and biomedical research can also be used to help us understand the factors that are leading to the extinction of species in the wild. By combining my knowledge of these laboratory techniques and my training in ecology and evolution, I have developed methods that can make a real difference for preserving species in their natural habitat. So the study I'll be talking about today is a good example of that. And I'm going to start out today by introducing you to a farmer from southern Ghana. This is a farmer that's farmed just outside the confines of the national park that I worked in. So you can see right behind him is the border of Kakum National Park. Now down here at his feet are his ruined manioc plants. He'd planted these so that he could feed his family for the next year, and he would have some of this to sell at the market to buy the things that he needed besides just the manioc. Now, overnight, he lost a great deal of his income and a lot of his potential future for the next year because forest elephants came out of the forest and both trampled and ate his crops. What you probably can't see is right down here at his feet is an elephant dung pile, which identifies the culprit. Now, he's really upset, and you can understand why. He believes there's too many elephants in that park, and if there weren't, they wouldn't come out of the park at night and feed in the fields. He'd like to see the size of the elephant population at Kakum reduced. And we can all sympathize with him, but let's put ourselves now in the place of the wildlife department. They know that elephant populations across the continent have been declining for the last 100 years, and dramatically so. They know that there could be other explanations for these elephants coming out of the forest and feeding in the fields. So what they need to know is how many elephants are actually in this forest, and how does that number of elephants, in other words, that density of elephants, compare with other forests of similar size. That way they can make an educated guess as to whether or not this population is too large and possibly should be reduced. Now what you can do if you know what your population looks like, if you can actually see your animals, is you can use traditional methods of counting animals. And this is a savanna population that's in a swamp, you can see here, in Mole National Park in northern Ghana. Traditionally, the method of counting these animals might be something like a line transect method. What you'd do is you'd map out your national park and you'd select about 6 points, we'll say, and you'd fly some line transects starting at that point and going, say, due east from there. You'd put 2 observers in your plane and they would look out either side and estimate the number of elephants in the park. Now, your map might look something like this. Here's your swamp down here, and here's your points here, 1, 2, 3, 4, 5, and 6. And here's an example of, say, a 10-kilometer line transect that you'd fly. Now, using the information from these line transects, you could use a relatively simple mathematical model to estimate the number of elephants in the entire national park. But this simply doesn't work when you go into the rainforest. You can imagine you can't see yourself hardly there. You can't see your people behind you on the trail, much less elephants. So what people have normally done in this situation is to use what's called an index. And an index means that you count something else and then you use that to estimate the number of, say, the animals that you're interested in. Now, the index of choice for forest elephants up to this point has been what's called the dung count. This was developed in its final form by a visiting scientist right here at UCSD, Dr. Richard Barnes, and his index relates the population size to the number of dung piles detected along transects. His way of doing transects is to pick a point in the forest from a random number table. Then he cuts a transect through the forest and counts the number of dung piles on either side of this transect for a certain distance. He then corrects that count for the dung deposition rate, in other words, the number of dung piles an elephant leaves per day, and the dung decay rate, that's how fast they stay, how fast they disappear from the trails, and finally for rainfall in the 2 months before the survey. Now, this last thing may seem a little counterintuitive. What could rainfall have to do with anything? But if you'll think about it, rainfall affects the amount of water that's in the plants that the elephants eat, which is going to affect the deposition rate. And it also affects the amount of water that's on the ground where the dung is laying, so that affects the decay rate. So it's an important parameter, and it really helped him to make his counts much more accurate. Now, he's also done a study which has looked at these counts when you can compare them with some other method of counting. For instance, in the savanna, if you did a dung count and you compared it with an aerial transect count, then he finds that the dung counts are very accurate. But what the big problem with them is they don't give you any information on other things that you need to know in order to effectively manage your population. And those are things like the sex ratio, just the numbers of males and females, the age structure of the population, or the genetic diversity, which we know is important if these populations are going to be able to adapt to future environmental changes. So the site that we did this work, and we each did a study at the same time, so they're companion studies, was Kakum National Park, and that's in Ghana. Ghana is over here in West Africa. The park itself is in the southern region in the deep rainforest area. Here's a aerial photo of this, and you'll see this park is absolutely isolated. The dark things that you see here are rainforest intact. Everything else around it has either been converted to agriculture or to human villages. Now, these fragments around the park do not any longer have elephants in them. So this population within here is truly an isolated forest elephant population. Now, we did 2 studies. He did 2 studies during the same year in the wet and the dry season using the dung count, and I did a genetic census in the same year. But before I could do my genetic census, I had to develop molecular markers to be able to do this. So I spent a year or 2 before this in the laboratory, and as Dr. Woodruff said, censusing forest elephants is a very dangerous task if you're going to have blood or tissue samples. So what I did was to develop non-invasive means of censusing forest elephants. And you can guess what that's going to be. We developed methods of extracting DNA from dung. Now, dung is going to contain the epithelial cells that are shed by the elephant as the food is passed through the gastrointestinal tract. But it's also going to contain fecal bacteria and potentially, if it's been on the ground for more than a couple of seconds, fungi and things like that. So I extract complete DNA, all of the DNA out of that, and I've developed some markers that are very specific to African elephants, so they'll help me to amplify only the elephant DNA out of it. Now before I leave this side, let me show you these things. See these round things here? Singular of this is bolus, the plural form is boli. Boli are how the dung falls, and they've been shown to be directly related to body size in elephants. And body size is directly related to age. So what we can do is by measuring the circumference of these boli, we can estimate indirectly the age of the elephant. And that's going to become important in a little bit when I start talking about what you can do beyond just a count with this. So keep that in mind. Now, microsatellite DNA markers, which is what I use to identify my individuals, are nothing more than simple tandem repeats of DNA. And they can be dinucleotides, such as these CA repeats. They can be trinucleotides, such as these TAA repeats, or tetranucleotides, like these ATTTs. What I used were the dinucleotides because they are very commonly found in the genome. And what you can see here are what are called alleles of those. Alleles are simply different numbers of tandem repeats. So this one here is a CA5, this would be a CA6, and this is a CA7. Now an individual inherits a single allele from each parent. So when you go to your individuals over here, you'll find that individual number 1 has inherited 2 CA5s, one from each parent. Now these are indistinguishable in size from each other. So when I use the polymerase chain reaction to amplify this individual's DNA, what I get on a gel is a single band because these two alleles are exactly the same size, and what this gel down here would do is to differentiate these these alleles by size. Individual number 2 has inherited a CA6 and a CA5, so you'll see here you have a larger band and then a smaller band there. So differentiated by size, you can see that this individual is what you call a heterozygote. This individual is a homozygote because both of his alleles are the same size. This one's a heterozygote because they're of a different size. So you can do this now for a number of different loci, and I did 7 loci, and I used the patterns at each one of those loci to compile what I call a genotype. Then you can use those, by your genotype, you can differentiate your individuals, because you wouldn't expect an individual, unless it's really, really closely related to another one, you would expect that at least one and probably more of these loci would be different between different individuals. Now I also wanted to know the sex of the individuals, because I was interested in the sex ratio. So what I did was to use 2 different markers together. This is the aldolase intron, and it's actually 2 introns. And what you see here is that if you amplify this in males and females, you see exactly the same pattern. There's no difference between the sexes and otherwise. What you see here, though, is this is a fragment of the Y chromosome. In mammals, that would only be in males. So you would see that a male would amplify a single band of the Y chromosome, whereas a female wouldn't amplify anything. Now if you put these 2 markers together into the same reaction, what you can do is differentiate between males and females. See this first one here, amplified only the aldolase introns. The second, third, and fourth, however, amplified that Y chromosome marker. So what you can say is, this is a female, these are males here, and all the rest are females. So with these molecular tools developed, I was able to go into the park, and my goal was here to sample as many dung piles or fresh dung piles as possible. So when I went into the park, I divided it into 15 approximately equal, what I'll call quadrats here, using latitude and longitude minute lines. The goal was to go in at the border, approximately say here, use a handheld GPS to roughly navigate close to this border here of this line, and then go north along there, and then back out. We used elephant trails as much as possible to avoid having to cut our way through the forest, and collected as many dung piles as we could in each one of these quadrats. We spent one day, and it was a field team of approximately 5 people, by the way. We spent one day in each quadrat and collected as many samples as possible. And I'll pass around this GPS so that anybody who wants to look at it can see what we were using. Here's an example of one of my field assistants collecting. And in this guy's hand, I didn't have a slide, I'm sorry, of him measuring the boli circumferences, but he has the tape measure there, and there's 2 boli there that my field assistant's collecting from. After they've been measured, he collects approximately 20 grams of DNA and puts it into a tube using a plastic fork. Now, it's really important that he doesn't touch any of that, so he's really very careful not to do that. I have here some samples of this. I'm going to ask you not to open these tubes because I don't want you to get the preservative on your hand. But here's a sample of these. You can pass them around and see what elephant dung looks like. Thanks, Taylor. So, here are the results here. And by the way, here is a gel that I ran. This is an example of a gel. And I'll let you look at it. It's not quite as pretty as what you saw over there and what you would expect those to look like, but you can clearly see the bands in this. So anybody who wants to look at it can. It's a little hard to see in this light. Lots of show and tell today. So we collected 205 samples in the forest. We successfully extracted DNA from 147 of those, and I successfully genotyped 124 of them. Now, this is pretty typical. When you're using a suboptimal DNA source, you do expect to have a little bit less success than what you'd have if you have perfect DNA, say, from a very freshly collected blood sample. So of those 124 samples, I determined that 86 were unique genotypes. The 7 loci plus the sexing all agreed on those that they were unique. But 38 were not unique. They matched one of those 86. So the challenge was to take these 86, what I'm going to call captures, and these 38, what I'll call recaptures, and determine how many elephants were actually in this forest. We know it's going to be more than 86 because I couldn't possibly have sampled everybody. So I used what's called Mark-recapture analysis. Let me show you how this works. Say this is K88, which was a male. He was captured, or his genotype was captured, in quadrat 1, quadrat 2, and quadrat 5. Another example is K321, a female. She was detected here in 5 and in 9. Now what you do is to compile a capture and recapture history for each one of these genotypes. So for K88, a 1 is going to show you that we captured him, and a 0 is a non-capture. So we'll put down each one of the quadrats here, and he'll have a 1 in 1, 2, and 5, and zeros in all of the other quadrats. Same thing, 321, she'll have a 1 in 5 and in 9, and zeros everywhere else. Now what I'm going to show you is a very simplified Mark-recapture type analysis. I have 15 capture sessions, and I have some things that are unique to my type of analysis. But Mark-recapture is an old analysis method, and here's how it generally works. It says that the number of individuals that you mark and release on your first capture occasion, in other words, the number of genotypes that I would detect on my first capture occasion, are related to the total population size in the very same way that the number of marked individuals you capture in your second capture occasion is related to the total number of captures in that second occasion. Now, this is oversimplified, as I said, but it's the general idea behind all Mark-recapture analyses, and this is often thought of as the Lincoln-Peterson equation. So my results tell me that I have 225 elephants in Kakum National Park. The confidence interval for that is about 173 to about 308. These are the results from the 2 dung counts that were done by Richard Barnes and his team. They estimate that there's 233 elephants. They have a little bit wider confidence interval than I have. But the fact that these 2 different methods, totally independent measures, give you almost exactly the same count means that we can have some really good confidence that we have somewhere between 200 and 250 elephants in this park. But this is where the dung count stops and the genetic census can go forward. I can look at the sex ratio. Using my sex markers, I determined that there were 35 males and 51 females in that population. And then remember those boli circumferences that we took? We can use that to look at age structure in the population. And this is a rough age structure. This is not an exact. This is going to be an approximate. So what we did was to say that the boli circumference of about 32 centimeters would correspond to an age of approximately 10. 10 is about the earliest you would ever see any reproduction in an African elephant. So we're calling those individuals with a bolus circumference of 32 centimeters or less juveniles, and all others as adults. So what you can see is that we have about an 80% adult ratio, and about 20% of the individuals are juveniles. What this tells us is that this is likely, at least over the short term, to be a self-sustaining population. That's something you wouldn't know using only a dung count. Now, I'm going to go on and try and explain these terms a little bit, but I think earlier you understood the idea that heterozygotes are individuals that have two alleles of very different sizes, different enough so that you can detect them in your gel. Population genetics theory predicts that if you have a randomly mating population, that you would have a certain proportion of individuals that are heterozygotes within that population. What we can do is look at the expected heterozygosity for the adults and for the juveniles, and we know that over something like 3 generations, because that adult group is going to include several different generations there, we know that that change that happens between these will have happened over that period of time. What we can estimate is that we've lost about 9.1% of the heterozygosity of this population over that about 3 generation interval. Now, we can also look at the alleles, the allelic diversity, and we know that rare alleles may very well be the genetic diversity that allows populations to adapt to environmental changes. So we will be concerned if we see a loss of rare alleles. In the adults, we have approximately 45 alleles. That's what I detected in my sample, whereas in the juveniles there's only 36. That difference is 9 for about a 20% allelic diversity loss. The other thing to note is that those 9 alleles were all rare in the adults. So that we're detecting there a loss of genetic diversity over these 3 generations that may signal that we are losing genetic diversity and that this population is suffering the effects of isolation. Now, so that you don't just think that this is something that has to do with a single elephant population in Ghana, let me put this in a bigger perspective. In 1989, there was a ban, a complete ban, on the sale of ivory and elephant products worldwide because of the declining elephant populations across the continent. The decline was so drastic that it was deemed necessary to put in a complete ban at that point. Now, in 1997, 3 southern African states petitioned to be able to list their populations as less endangered and to have a very limited sale of the ivory that they had stockpiled from culled elephants during that time. Now, many people were concerned that this would result in increased poaching across the continent and that African elephants way outside of these 3 states would be affected. What we needed were good counts of elephants from all the way across the continent of Africa. But at least a third of the African range states have not had recent surveys and therefore reported to the African Elephant Database what are called informed guesses. This means that we don't have good information from before for a lot of these range states. Now, I believe that my genetic censusing method can help to alleviate some of the problems based on this lack of information. If we do genetic censuses on key populations and dung counts on surrounding populations around them, because they are a little bit cheaper to do than the genetic census, I believe that we can look at the variability within the populations, and we should be able to look at the future trajectory of these populations and find out if they're still self-sustaining, if we've lost the majority of the males, these type of things that we'll need to know to detect the effects of the ivory trade on these populations. Now, before I leave, I'd like to tell you that funding for this project was provided from 2 sources. First of all, by a grant from the UCSD Academic Senate to David Woodruff, and also by a grant from the African Elephant Conservation Fund, which is administered by the U.S. Fish and Wildlife Service. Thanks a lot, and I'll be glad to take questions if there's time. That's a really good question. She was asking if my census seemed to indicate that there were more females than males in the population. And when you statistically test that, it says that there's not. However, that's very, very close. The P is about 0.06 for that. So what I think is that it's possible that if there isn't a difference, it could be the results of my sampling method. And the reason for that is that I was following elephant trails through the forest. Well, elephant trails are real obvious when it's a large group of elephants. And we know that the matriarchal structure of African elephants, at least savanna elephants, says that the females and their young will be traveling together in groups. So by following elephant trails, I may have missed some of those males, especially the lone males in the center of the forest. So it's definitely a potential that we're still seeing the effects of poaching from before, but it also could be the effects of my sampling method. The first question is, if we were to count all the elephants in Africa, what would the cost be? And then the second one was, if the elephants were leaving the forest and there were other reasons for it, what might those reasons be, other than just coming out and cooperating there? The first question is, the cost would be tremendous if we had to do genetic censuses for everyone. So my proposal would be to only choose key populations, those that we really think we're going to be able to put money into conserving, and that might be national parks that are actually giving back to the communities. So I would say that those populations we should do genetic censuses. Other ones, I've shown, and it's been shown repeatedly, that the dung counting method works. It's good, it's accurate, and you can do it repeatedly, say at 2-year intervals, to look at changes, and you should be able to detect trends that way. It's also really good that African personnel can be trained, just as they are being done in Ghana now, to do these in their own countries, and they can involve their own range scientists. So I think that's important. Okay, so the second question was, what are the reasons that the African elephants might come out of the forest? And they're real complicated. The first one involves the fact that you saw that he was right there on the edge of the forest. Being right there on the edge of the forest is kind of like putting candy out for a baby. You could be actually drawing them out by putting palatable type crops right on the outside of the forest. The other thing could be that along the edges of the forest there are the last logging areas. So when the loggers leave the forest, they leave the center first and they log towards the outside. African elephants really like good leafy green vegetation. Recently logged areas have a lot of that. So we could be drawing the elephants to the edges of the forest right next to the fields simply by our logging practices. So that's another one. So the third one, of course, would be that there isn't enough food in the forest. What we'd need to do to document that would be to look at food plants, look at their distribution in the forest, and find out whether or not there indeed may not be. Are there cash crops that we can put on the outside of the forest that elephants are not particularly attracted to? And the best thing there is something that I hate to recommend, and that's tobacco. They absolutely hate tobacco. They don't like to smell it, but I really hate to see Ghanaians go into the tobacco business, so it's hard to recommend that. Now in this particular area, the elephants for some reason don't take cocoa. They do in other areas, but for some reason in this park they seem averse to it. So it's another one that you can put there. Cocoa's more of a tree form. It's a little harder for the elephants to get around through, and if you plant it densely, they may have a little more trouble. So that may be part of that. That's a good question. The question is it being used on other species, and what type of animals is it being used on? Well, the first type of study like this was done on whales, if you can believe that. They slough skin. And the observer boats could go behind and pick up these big sloughed pieces of skin and do DNA on that. That's amazing. You can also use biopsy darts, same kind of thing. But they estimated, it's Per-Paulsbo's group, and they estimated, I believe it's the right whale population in the North Atlantic. I'll have to find out about that. But that's really interesting. It's been used on mountain lions, it's been used on bears, especially a population in Europe called the Brenta bears that are down to about 5 individuals. So it's been used on them. It's used on grizzly bears. It's used on coyotes. A group up at UCLA estimated the number of coyotes in a particular region using that. So it's been used quite a bit. I'm the first one who's used it on forest elephants, but I'm certainly not the first one who's used it, although I'm up at the head of the pack, kind of. So, well, thank you very much for coming. I appreciate it.